Hey there, this is Dr. DiMaio again. This is your final notes for Endocrine System. I want to just summarize everything. We're going to be talking a little bit about some interesting things called Ecosonoids. And if you remember from A&P1, you learned about different types of lipids in the body. Ecosonoids are one of them. And by the way, it happens to be a hot topic during this COVID pandemic, where people were actually going into cytokine storm, and that's what triggered a lot of inflammation and then the subsequent coagulation and blood clotting and problems with oxygen deprivation. As we get into the blood, you'll see why if you damage the hemoglobin and you damage the red blood cell, you will not get good oxygen saturation. So this is about endocrine, and this is about your health. So let's look at this. So this was in a note form in Word document. I put it into this PowerPoint so I can make this audio narration for you. The endocrine system, regulates many functions in the body. It does work hand in hand with the nervous system, and we call that aspect the neuroendocrine, right? So we have neuroendocrine system, basically. It's a combination of the two. We sometimes refer to many functions as the neuroendocrine because of its intimacy. The hypothalamus regulates the pituitary and its tropic hormones. The posterior pituitary is an extension of the hypothalamus. It has an area called the neurohypophysis. This one is facing, that's anterior, and that is posterior, right? So this is the front, this is the back. So this is actually the anterior pituitary gland on this image, and this is the posterior pituitary gland, right? And so you're seeing nerve fibers coming down to the posterior pituitary gland, and you're seeing glandular tissue in the anterior pituitary gland. That's why sometimes they call it the adenohypophysis, because adeno prefix means glandular, right? Now you could probably see this better in the notes that I already gave you. You have the actual Word document. It's kind of small in this PowerPoint because I'm making the recording. I did add this little image here of the Lamprey Eel, and this is what we dissected at NASA in comparative anatomy. And actually, I got this off the internet, and it's from a site where they sell these for labs, right? Now, you don't see his eyes too much. See, the eyes are kind of covered on this eel. There's different types of lampreys, just to let you know that. This is a deep water lamprey. And then on the top, you can't really see it, but there's actually a lens. There's no real lens here. It's covered, but there's a lens on the top. And then there's another eye that's covered on the other side. Underneath this lens is the pineal gland on this species. So the pineal gland is important because it helps to regulate the circadian rhythms of the body. There's a lot of weird stuff on the internet. People talk about the third eye and all this stuff. They get into spiritual stuff. But the pineal gland is this brain structure. It's part of the endocrine system. It receives neural input from the eyes when exposed to sunlight. Since he doesn't have eyes, he has a lens like an eye right above it, and just below is the pineal, so you can get direct light. Because this eel lives at least 500 feet underwater, and there's very little light, but light does get there. And so it receives that light, and the UV reception stimulates the pineal gland to release melatonin. Here's melatonin here. Look at the structure of the melatonin. Look at the structure of serotonin. Very much related. So it's important, and so important is sunlight, that the sunlight-derived neuroendocrine pathway, that it can mean life or death to a depressed person. It could mean life or death with COVID as well, because if you're not getting enough sunlight, we know we're not going to make enough vitamin D in our skin as well. The relationship is in the chemistry of the melatonin, in this case for the depression, and the similarity to serotonin. Also, the production of serotonin is increased from sleep that the melatonin induces. So the melatonin induces sleep. When you get good sleep, you get a better production of serotonin, And serotonin helps you to recall better, besides help your depression and a feeling of satiation. You know, serotonin is known as the feel-good hormone. You're just satisfied and happy, you know. But melatonin puts you to a nice deep sleep. When you get deep sleep, you will produce enough serotonin. If you produce enough serotonin, then you have good recall. And as a student, you will do well if you get sunlight in the daytime through your eyes. I'm not talking about looking straight at the sun. I have to repeat myself on that one. You never want to look directly into the sun, but you don't want to be outside all the time with UV glass covering your eyes, whether it's the sunglasses or your car or your house. People think they could sit in a car or something with UV glass. You're getting sun into you're not getting it because the UV glass disperses it laterally and it doesn't penetrate. So you need to have sunlight in your eyes for this production of melatonin. And the more sunlight in the daytime, the more melatonin you will produce when the sun goes down. The melatonin helps you to sleep deeper. The deeper you sleep, the more production of serotonin. The more production of serotonin, the better you feel, and the better you recall. Interesting, huh? And here is a summary of endocrine-related effects of sunlight. And I have references if you want. It's 2 pages of references. It was just too much to put here. I tried to scan it from my book. Well, look, it lowers, when you're in the sunlight, it lowers your blood pressure. It lowers your abnormal high blood sugar. If you have diabetes, it increases your sex hormones, increases your body's tolerance to stress. It lowers resting heart rate, lowers your cardiac output, lowers blood cholesterol. That's nice. It increases energy, endurance, and muscle strength. It increases the body's resistance to infection, and it increases the oxygen carrying capability of your blood. That would be good. That would be a good thing, especially with COVID, right? Because that's the problem. And it decreases the size of your internal cancerous tumors. Yep, I said it. It decreases the size of internal cancerous tumors when you get sunlight. All right, if you need that reference, you can ask me. I'll let you know. Okay, also, there's a space where you got to realize that the hypothalamus extends down. You have an infundibulum, the stalk of the pituitary gland, so to speak, which is also the septa, or the separation between the anterior and the posterior. And so the infundibulum of the pituitary gland, or the septa area, or the central part of the pituitary gland, is where you produce the melanocyte-stimulating hormone. It is in response to sunlight entering the eyes, and within the brain, the substantia nigra, there are also pigmented cells. Now look, your eyes have pigment. You have the color of the choroid plexus, and then you have the pupil, right? And so the pupil is pure melanin, right? So you have this melanocyte type cells. Then you have within the brain melanocyte type cells in the area called the substantia nigra. And they're pigmented cells associated which produce dopamine. And dopamine is an amino acid or peptide-derived neurotransmitter that affects your brain, and it has to do with movement. And Parkinson patients have problems with L-Dopa. Dopamine helps with the mood and the reward cascade for addiction as well. So maybe we'll find out in the future how sunlight can help Parkinson patients. I do know that you could take adrenal medulla cells and transplant them into the substantia nigra because they all come from the same origin. Melanocytes, neural crest cells is where melanocytes come from, and so do the adrenal medulla cells. So they all come from the same precursor cells in the developing embryo. And if you transplant adrenal medulla cells, which originated in your same area where your nerves originated, and they start to convert and they make dopamine. It's pretty amazing. They did research on that. So again, melanocytes are from the same origin, embryonic origin, as neural crest cells and as our adrenal medullary cells. So there is a relationship with your nervous system. Thus, our mood, our sleep, possibly even our motor functions can be helped through regular sunlight. And that's without UV glass or sunblock, don't forget that. As we learned in AMP1, vitamin D can only be synthesized when our skin is exposed to sunlight, and that vitamin D is necessary for calcium absorption. And it's also known that calcium ion is the #1 gene regulator, and we will see this movement of calcium ions in the nucleus in some cases as the second messenger system. And the calcium ion actually can be a second messenger. We've seen the images uploaded in the PowerPoint, the locations of the endocrine glands and endocrine tissues. We've also discussed what it means to be endocrine, and a product that's produced by a cell or cells in one area of the body, usually surrounded by lots of capillaries. And there you'll see they're specialized capillaries. We talked about how they're porous, right? And the product enters that bloodstream and travels to a distant target cell. Whereas an exocrine gland has cells that line a lumen of a duct, and the cells produce a product, enters the duct, and pours out on a surface, such as your sweat or oil glands. And it actually should say lipid-soluble hormones, because we don't just have steroid hormones. We have another chemical I originally started talking about. It's called the Ecosonoids, and they are lipid-soluble as well. So I want you to, in your notes, to write lipid-soluble hormones above the steroids. So amino acid derivatives such as tyrosine is used to make thyroxine, epinephrine, and foods high in tyrosine include meat, dairy, fish. Tryptophan is specifically used to make melatonin. Foods high in tryptophan include turkey, chocolate, oats, bananas, dried dates, milk, cheese, and peanuts. And when you eat a lot of tryptophan foods, it helps you to sleep better and even have better dreams. There was actually a bodybuilder named Frank Zane during the time of Arnold Schwarzenegger's era, and he was really in good shape, but he used to tout and write articles about the benefits of taking tryptophan before you go to bed. You used to be able to find tryptophan as a separate amino acid very easily. Now it's very difficult to find it because people were overdoing it. Because tryptophan, if it helps you to sleep, guess what it helps besides? If it helps you to make melatonin, guess what else tryptophan might help? Anybody know? Like I'm waiting for you to answer me. It helps to make serotonin. And serotonin is important for feeling good, feeling satisfied, and memory. Pretty cool, huh? So he used to actually talk about almost like taking a tryptophan trip, like tripping out on tryptophan, because he was able to get into some really cool dreams, So turkey, we all know if you eat turkey, it helps you sleep better, right? Okay, so peptide hormones would be considered, what's a peptide hormone? Instead of amino acid is more smaller than peptide is a chain of amino acids. And most start as prohormones that are inactive but become activated after they're secreted. A good example is glycoproteins, and these are really important on the cells, generally speaking, but they're about 200 amino acids or more. And that includes your TSH, LH, and FSH. And then there's short polypeptides or small proteins. 9 amino acids long would be ADH and oxytocin. 191 amino acids long is growth hormone. And 198 amino acids long is prolactin. This group includes all the hormones released by the hypothalamus, the thymus, the digestive tract, pancreas, posterior pituitary gland, and several other organs in the body. It's a big group of short polypeptides. Then we have the steroid or lipid-soluble hormones. The steroids are one, and they're released by male and female gonads, and also the adrenal cortex, and even the kidneys release calcitriol. They will bind to a specific transport protein in the plasma and stay in circulation longer than peptide hormones. So hormones can modify cell activity, obviously, and we're talking about metabolism usually, like altering the membrane permeability triggers things to occur. It activates enzymes. We said enzymes are necessary for every metabolic chemical reaction in the body, and it even can affect genetic activity. cells must have a receptor specific for the hormone to affect it. Each cell has receptors for different hormones. Not all have the same arrangement or same receptors. Thyroid hormone affects just about every cell in the body. And hormone receptors can be on the cell membrane or inside. The receptors of the catecholamines, dopamine, norepinephrine, remember, these are not lipid soluble, so they're going to be on the outer cell membrane, whereas the ecosonoid and steroid receptors are on the inner surface of the cell membrane or inside the cytoplasm. Remember, ecosonoids are fat soluble and can pass right through the cell membrane. Thus, there are extracellular membrane receptors and that means inside, intracellular membrane receptors. Hormones cannot directly affect the function of the cell. They must use a second messenger, which is an intracellular intermediary. And there's usually a bridge to that, which is the G protein. So these are examples of 3 different types of second messengers: cyclic AMP, cyclic GMP, and calcium ions actually act as a second messenger. So sometimes you can have this thing called amplification, and that's when a small number of hormone molecules bind to receptors. Thousands of second messengers suddenly appear. Then you can have receptor cascade, when a single hormone binds to the receptor more than one type of second messenger, or production of many linked sequence of enzyme reactions occurs, and this can affect many functions of the cell at once. And then you have this thing called permissive effect. Permissive effect is when a hormone cannot perform its full effect without the presence of another hormone. And then we have this thing called synergistic effect, and that's when 2 or more hormones have the same effect on a target cell and it amplifies it. Not to be confused with amplification, right? Because amplification is when you have a small number of hormones bind to receptors and all of a sudden thousands of them appear. A synergistic effect is when 2 or more hormones have the same effect on it, and it causes a stronger effect on that second, a stronger effect on the cell. Then we have this thing called antagonistic effect, and that's when a hormone reverses the effects of another hormone, such as glucagon, doing the opposite of insulin. And then we have downregulation and upregulation. Downregulation is when there's a presence of hormone at the cell more than usual. It causes the shutdown of receptors and the cell becomes less sensitive. Upregulation is when the levels of hormones are lower than usual and the cell increases its number of receptors, making it more sensitive to the hormone. So prostaglandins and hormones. What the heck is a prostaglandin? A prostaglandin is a type of ecosonoid. It's under the heading of ecosonoids. Well, they were first called prostaglandins because they found them in the prostate of males. Yet we find these chemicals all over the body in both males and females. And this article, I was trying to find it again. I had copied this years ago, and I guess they changed the link for it. It was an article written by Barry Sears. So I guess you'd have to go through all those articles to find this. But basically, I took some information out that's very powerful and helps us to understand this really well. The word ecosonoids derived from the Greek word for 20, which is ecosa, since all of these hormones are synthesized from essential fatty acids that are 20 carbons in length. The first actual ecosonoids were discovered in 1935 by Ulf von Uhle. These first ecosonoids were isolated from the prostate gland, as I said, and an exceptionally rich source of ecosonoids. And they were called prostaglandins, a small subset of a larger family of ecosonoids. Since it was thought at the time that all hormones had to originate from a discrete gland, it made perfect sense to name the new hormone prostaglandin. And with time, it became clear that every living cell in the body could make ecosonoids. Did I tell you that? Did I say it right? I don't know. It became clear that what? Every living cell in your body can make ecosonoids. And there's no discrete organ. It sounds like a test question, right? Or gland that was the center for ecosonoid synthesis. To date, biochemists have identified over 100 ecosonoids and finding more each year. The breakthrough in Ecosonoids research occurred in 1971 when John Vane finally discovered how aspirin, listen to this one, the wonder drug of the 20th century, actually worked. How does it work? Well, it changes the levels of eicosanoids. It literally blocks all prostaglandin production. Now, you may not understand what that really means. It's actually not a good thing all the time, especially with you trying to fight off a virus. The 1982 Nobel Prize in Medicine was awarded to Vane and his colleagues, Bengt Sandelson and Sun Bergelsen, for the discovery of how eicosanoids play a role in human disease. He further states, eicosanoids are hormones that maintain the information fidelity of your biological internet. Ooh, that's a pretty word, thing. Which means they can become the key to health and longevity. Why are eicosanoids so unknown if they're so important? First of all, they are made and act and they self-destruct within seconds. That makes them very difficult to study. Secondly, they don't circulate in the bloodstream, making it extremely difficult to get a sample. Finally, they work at a vanishingly low concentrations, making it almost impossible to detect them. Despite these barriers, more than 87,000 articles on eicosanoids have been published in peer-reviewed journals. So the basic research community is interested in eicosanoids, even if your doctor never learned about them in med school. Eicosanoids encompass a wide array of hormones, many of which endocrinologists never heard of. They are derived from a unique group of polyunsaturated essential fatty acids containing 20 carbons. The different classes of eicosanoids are shown below here. You have subgroups of eicosanoids. Prostaglandins are one, thromboxanes are another. Look at the prefix, leukotrienes are another. Lipoxins, hydroxylated fatty acids, aspirin-triggered epoxins, isoprostanoids, epoxy-eicosanoic acids, and endocannabinoids. I'm sure that's familiar to some of you, cannabinoids, right? Dr. Sears further states that there are other eicosanoids as mentioned in the textbook, such as leukotrienes. As the name implies, leukotrienes are produced by white blood cells. We're going to learn about leukocytes soon, and are involved in immunity. Also, cytokines are similar but come from other different types of cells. Dr. Sears says we can alter our eicosanoids to shift towards good or bad depending on what we give the body. There does remain one way to directly manipulate eicosanoids, and that is your diet. You must realize he did all his research on drugs and eicosanoids. Dr. Sears also says the cell, the basic structural functional unit of the body, has the resources of fatty acids within the cell to make the eicosanoids depending on the cell's environment. And I said, this is a repeat of that slide, that the cells will test their environment, test its own environment with an eicosanoid. It's like popping smoke in a combat situation and then seeing how they react to it, or putting up like an old cowboy movies. They hold the flag up or a helmet up and they shoot through the helmet, right? And they want to see where the round is coming from. So as endocrine hormones, eicosanoids' mission is to be secreted by the cell to test the external environment, report back to the cell what's going on and how to respond to that. And the way it responds to it is by making a specific second messenger based on what stimulus is coming towards that cell. Why does this have such an effect? And why is it so important? The reason why eicosanoids play such a central role in our physiology is due to the second messages that certain eicosanoids generate. There are a variety of eicosanoid receptors on the surface of the cell, and depending on the eicosanoids, on which eicosanoid interacts with the receptor, a specific second messenger is then synthesized by the cell. And sometimes a second messenger such as cyclic AMP is generated. Sometimes a totally different one can be generated, such as DAG or IP3 we saw before. And if one second messenger goes up, then the other goes down. So in essence, the complexity of your biological internet is reduced to a digital system consisting of green and red lights. Those ecosonoids that generate increased production of cyclic AMP are your key to maintaining wellness. Let us discuss the second messenger system, cyclic AMP. And first of all, the link between the first messenger and the second messenger, we said earlier in the other video, is the G protein. Remember that. The first messenger is the hormone itself. The G protein is an enzyme complex linked to the membrane receptor. The G protein can bind to what's called GTP, which is a nucleotide-based high-energy compound that can transfer energy in specific enzymatic reactions. The text tells us that about 80% of prescription drugs target receptors coupled to G proteins. This activates the enzyme adenylate cyclase, thus converting ATP to cyclic AMP. The cyclic AMP now functions as the second messenger, and it typically acts upon the enzyme kinase that phosphorylates proteins. Phosphorylation is when you're attaching a high-energy phosphate group to something. The phosphorylation of membrane proteins can open membrane channels, and when the liver and skeletal muscles release glucose from stored glycogen, that's what's going on. Also, hormones such as thyroid-stimulating hormone, glucagon, FSH, LH, ADH, ACTH function in that same way. It is a short-lived due to enzyme called PDE. So this actual production is short-lived because PDE will shut it down. Those ecosonoids that generate increased production are your key to maintaining wellness. Producing increased production means you're producing cyclic AMP. Cyclic AMP is the same second messenger used by a number of endocrine hormones. And by maintaining adequate cellular levels of those ecosonoids that increase cyclic AMP, you're guaranteed that a certain baseline level of cyclic AMP is always present in a cell. Thus, it's far more likely that the overall cyclic AMP level in a cell will be high enough to ensure that appropriate biological response. And that means you'll have better hormonal communication. How can you tell a good from a bad ecosonoid? Ecosonoid's effect on a second messenger becomes the definition of good or bad. A good ecosonoid will what? What will a good one do? Increase levels of what? Cyclic AMP. Whereas a bad one, bad ecosonoid will decrease levels of cyclic AMP through the elevation of IP3 and DAG, second messengers. So the table shows you a listing of types of good and bad ecosonoids and their receptors. Receptors for good and bad ecosonoids. So a receptor that affects on cyclic AMP is good. So PG1, EP2, EP4 increase. PGI2, IP increase. PGD2, DP increase. The bad ones are these. Okay. Now, notice this PG means prostaglandin 1. That's prostaglandin 12, prostaglandin 2. And this is prostaglandin, this is PGD2 and this is PGE2. So this is a bad one. And from what I remember, when you take medication for asthma, one of the medications is an adrenaline, norepinephrine type of thing to open up the airways, and the other one is a PGE2 inhibitor. And so you want to try to actually open up the airways. And that's what that enzyme does. It and reduces the swelling. So this produces swelling, this bad one, inflammation. And if you inhibit it, you block the swelling. That's how that medication works. So a lot of medications are designed, 80% they say, are designed to affect these ecosonoids in one way or another. So let's look at G proteins and calcium ions. A G protein can either open calcium channels within the cell or the release of calcium ions from other organelles in the cell. The G protein activates the enzyme PLC, phospholipase C, triggering a receptor cascade. We talked about that before, starting with the production of DAG and IP3, and then when IP3 enters the cytoplasm, it triggers the release of calcium from the smooth ER, and then the combination of calcium with DAG activates another enzyme called PKC, and leading to the phosphorylation of calcium channel proteins, allowing calcium to enter the cell, triggering a positive feedback loop. Now, as I'm talking about this, you can open up the next slide where it shows you that. I believe it's in here. So let's go back. So the G protein, what happens? G proteins, as a G protein, can either open calcium channels on the membrane or on organelles. The G protein activates an enzyme called PLC that triggers a receptor cascade, and starting with the production of DAG, IP3, and then the IP3 enters the cytoplasm, it triggers the release of calcium ions from the smooth ER, and then the combination of calcium ions with DAG activates another enzyme called protein kinase. In this case, it's called protein kinase C, leading to the phosphorylation of calcium channel proteins, allowing calciums to enter the cell, triggering a positive feedback loop, increasing cytoplasm calcium levels inside the cell even more, and then the calcium ions bind with a protein called calmodulin and serve as a second messenger that activates enzymes. This is common in the stimulatory effects of epinephrine or norepinephrine, and is also involved in oxytocin and some regulatory hormones. So once an ecosonoid interacts with its unique receptor, a second messenger is then synthesized inside the target cell. If a good ecosonoid interacts with the right receptor, then cyclic AMP is the second messenger. On the other hand, if a bad ecosonoid interacts with the receptor, then cyclic AMP levels are decreased. Adding further to that complexity, that some ecosonoids such as PGA and PGJ are cyclopentanine ecosonoids. These ecosonoids don't have cell receptors on the surface, and they can directly enter the cell where they can interact with the cell's nucleus to affect cellular growth and differentiation. Since there is no discrete ecosonoid gland, there's no central site that turns on or off ecosonoid action. Nature solved this problem by developing different types of ecosonoids that have diametrically opposed physiological actions. It is the balance of these opposing actions of different ecosonoids to remain in equilibrium of biological activity. These differences in biological actions are the foundations for what's called the ecosonoid axis. He further states, the 1982 Nobel Prize in Medicine provided me an insight into the molecular nature of chronic disease, since it could be seen as an imbalance in ecosonoid levels. It became apparent to me at the same time that the appropriate balance of ecosonoids could be used to provide a molecular definition of wellness. In essence, the more balance of ecosonoids is tilted toward bad ecosonoids, the more likely you're to develop chronic disease. Conversely, the more balance is tilted toward good ecosonoids, the greater chance of you achieving wellness and longevity. I got to say, we talk about this in my practice. We talk about putting somebody into a healing state. So the state of your body's health is more towards healing. And a lot of things will do that. Proper nerve supply, proper nutrition will do that. And how does proper nutrition do it? On a cellular level, one of the ways it does it is by affecting your release of different ecosonoids. If you eat something high in sugar, you're going to produce bad ecosonoids. And the production of bad ecosonoids actually lowers your energy to the point where it actually shuts down some of your mitochondria. So the amino acid and essential EPA ratio will indicate where you stand in terms of balance. If you're skeptical about this statement that ecosonoids play a fundamental role, then ask your physician what happens when they give a high dose of corticosteroids to a patient for more than 30 days. The answer will be physiological devastation, if not death. This occurs because corticosteroids have one mode of action. They knock out all ecosonoid production, good and bad. And they do it by inhibiting the release of the essential fatty acids from the cell membrane. So I may have mentioned that amino acids and essential fatty acids are necessary in your diet, and a lot of people do not take supplements for that. and you should. And essential fatty acids include, your fish oils, right? And amino acids should include your 9 essential amino acids. So what happens is if you throw off that ratio and then you block all your ecosonoids, good and bad, it's actually doing it by inhibiting the release of the essential fatty acids in the cell membrane. So this chokes off all supply of precursors to make any type of ecosonoid. Without ecosonoids, you can't survive. Dr. Sears is the founder of the Zone Diet, a diet based upon regulating the ecosonoid balance of good to bad, controlling insulin to glucagon levels, and balancing the essential fatty acids and amino acids in your diet. It revolves around a 40-30-30 balance of protein to carb fat, but the fats must include specific balance of omega-3 fatty acids and the omega-6. There are no feedback control centers for ecosonoids. There's no specific organs of ecosonoids. They balance each other. Corticosteroids will destroy good ecosonoids and have a profound effect on your health. Good ecosonoids increase cell energy, whereas bad ecosonoids slow down cell energy. The hypothalamus and the pituitary gland do not control ecosonoid production. Cells will use ecosonoids as autocrines and paracrines. The control of hormones other than ecosonoids, endocrine reflexes triggered by blood humoral stimuli changes in the composition of the extracellular fluid, hormonal stimuli, arrival of or removal of a hormone, and nerve stimuli, arrival of neurotransmitters or neuroglandular connections. Hypothalamus is the highest level of endocrine control. It's a feedback mechanism using negative feedback. The hypothalamus synthesizes hormones and transfers them along axons to the posterior pituitary, and then they can release into circulation. The only 2 hormones synthesized by the hypothalamus we know are ADH and oxytocin. The hypothalamus secretes regulatory hormones that control the release of anterior pituitary. We call those the tropic hormones. And when released, they have an effect on the endocrine gland to release their products. That's a typo there. To affect target cells. Hypothalamus has the control center for the release of adrenal hormones. The adrenal medulla cells and their hormones. And that does it through the sympathetic nerves. There's a sympathetic nerve from the spine that goes directly into the gland. And it's white, myelinated. That means it's very fast. It does not stop at go. It doesn't stop at a ganglia. So it's all preganglionic, having a synapse directly into the medulla. There's no synapse on the way.